we have Dina with more genetics.
Hi everyone again. So I'm going to give you a brief overview of my project today, and I work on adapting eDNA methods for monitoring intertidal biodiversity. So I kind of struggle to explain my project to a lot of my family and friends, so I like to use eDNA as a kind of real-life tricorder. And that is because, firstly, we can scan environments and different habitats. We can store this data and convert it into a digital format, and we can do analysis. And I really wish it was as easy as Spock makes it look in Star Trek. So I am an ecologist, and I work in intertidal environments. And a lot of coastal regions are extremely vulnerable to climate change, and they are very valuable. They offer a plethora of different ecosystem services. However, because of the impacts of climate change, such as ocean warming, ocean acidification, a lot of these species that live in these habitats have reduced fitness. And so their distributional ranges often shift and change over time. And because a lot of the intertidal species are invertebrates and algae, they have quite short lifespans. They're also ectothermic. And so this means that they're super sensitive to climate change, and we can actually use them as indicators for the ecosystem as a whole. So it's very important to monitor these species. One amazing intertidal biodiversity monitoring program is the Markland project. It is, to our knowledge, the most spatially and temporally extent monitoring program in the whole world. It's solely visual surveys currently, and it covers a wide range of invertebrates and macroalgae species. And its main goal is to detect these biogeographical shifts in across this kind of tree of life with climate change. So although these monitoring programs are super valuable, they also have limitations. For example, they often miss rare, cryptic, or small species, so we can actually undersample the biodiversity in certain sites. They also heavily rely on expert identification skills. Nova is amazing. She knows every single species on the shore, but she can't be cloned and be put in multiple different places all at once. And because of this, there is quite a lot of financial strain for visual surveys. So I'd like to pose the question whether these surveys can be complemented in any way. And so that's where I kind of come in. Over the last decade, there has been a rise in molecular methods. They've kind of been the talk of the town, if you will. And that's because they pose a lot of solutions to these problems. So I work on environmental DNA, and some might say, what is environmental DNA? So as the kind of animals and plants do their daily activity, they shed DNA into the water, and I can kind of scoop up that water and detect what DNA is in there. And this is done through multiple different processes, such as swimming, excreting, mating, and it's a soup of DNA in the water which I can collect and look at the species presence and absence. Now, the eDNA methods in marine ecology have become quite popular over the last decade, as you can see by the increase in publications. And one of the most popular methods for biodiversity is metabarcoding, and that's because it allows multiple different species to be detected in one sample. The way that metabarcoding works is just like in a supermarket. You scan a crisp packet or a cereal box, and these universal barcodes are how we detect what species are present and absent. And these barcodes are conserved across the tree of life, and so we can kind of pick them out, scan them, and understand what species are there. So applying these eDNA methods could really improve the way we monitor intertidal environments, and so that's what my project's looking at. The work in the intertidal is quite limited, and that is because it's a super dynamic and diverse environment, and it's very difficult to disentangle these spatial and temporal variables. So for the first two chapters of my project, I'm firstly looking to see the spatial capability of eDNA in rocky shores. And secondly, I'm going to do a comparison between the visual survey data and the environmental data to see how they differ and what they detect. And I'm targeting all of the Markland species that currently are on NOVA's kind of detection list. So it's, like I say, it's not like Star Trek. There's a whole long process to get through, from field collection to eDNA extraction, all the way through to kind of getting to the community analysis. And I've kind of done 2 batches over the last 2 years, and you can see my progress over 2022 and 2023. And so I'm kind of getting to the end of it now, and hopefully we'll have some lovely data soon. I have collected paired eDNA samples across many different sites with the visual survey data, with lots of helping hands. And I've collected 262 samples across 32 sites in 5 major regions of the UK. So I've done Scotland, Northumbria, North Wales, South Wales, and Cornwall. There's been a lot of driving. In terms of the spatial questions that I want to look at, I want to see whether the eDNA is capable of detecting diversity differences between these major regions and also between the sites within these regions. And so from the literature, we know that eDNA is very sensitive. And so what I do expect and hypothesize is that we're going to see differences between both regions and within sites. Going to more a local scale, I'm interested to see whether the eDNA can actually detect differences across the shoreline. So I've got samples from high shore, low shore, and open water to see whether we can actually detect succession across a shore. Again, because of the sensitivity of eDNA, I am expecting to see some differences. This is what it looks like when I filter my water on the shore. I use a very technical sealant gun to filter my water. The DNA is collected in this specialized Sterovex capsule, and it's taken back to the lab to do some lovely magical eDNA extractions and amplification so that then the sequencing machines can detect these sequences and assign them to the taxonomy. So we use different barcodes to obviously detect different species. So CO1 is for the invertebrates and 18S is for the macroalgae. It's all very much still a work in progress, but I do have some preliminary results for you, but take them with a pinch of salt. This is only a fifth of my data set that I actually have back right now. So we know that the protocols work actually quite well. The methods are cheap, they're portable, and the quality and quantity of the DNA that we're getting back is actually very promising. From the data analysis and the bioinformatics that I've completed so far, we're getting very sensible species assignments. So I'm just going to present some data from one of the barcodes, which is the 18S barcode, and just from 2 regions that I've actually got kind of in front of me. So these are all of the algal species that we've managed to detect over these 2 regions. As you can see, we're detecting quite popular groups across green, red, brown algal, and we've got over 140 unique species detected. When I look at the diversity differences between the regions, I use 2 different metrics. We can see that actually the eDNA is detecting a difference. So Scotland in the orange and South Wales in the blue, we're seeing that Scotland is significantly higher diversity than South Wales. When we kind of zoom in and look at this on a site level, we can see that there's loads of variation. And it's evidence to say that eDNA is very sensitive. And so it is likely that this method is going to be quite useful across large monitoring. However, when we do look at the diversity differences between sample types, so I've kind of grouped together the rock pools and compared the open water samples with the rock pool samples, we're not seeing much of a difference in diversity detected. However, what we do see is that the rock pool samples have a lot more variation, which makes sense because you can get a rock pool that's pretty barren and doesn't have many creatures kind of hiding in its holes, and then you can get another rock pool that is super diverse. So yeah, it kind of makes sense, but to actually disentangle these patterns a bit more, I have to have more time to kind of understand what's fully going on. So yeah, this could mean that eDNA has potential lower sensitivity on these kind of much finer scales, but again, I need to look a bit more into it. When I look at some preliminary results from a short pilot study I did for the invertebrates, we're finding that a lot of the species detected do match the visual survey. So the little icon with the eye and the tick are the species that we saw on the site and what the eDNA also detected. But we're also seeing that they're detecting a lot more species. I found there was a lot of variation between sites as well. So again, this is kind of linking back to the sensitivity of the eDNA. And some groups are more easily detectable than others, and this links to how the rate of which different creatures and different species shed their DNA into the water. So we had a difficulty with gastropods because they're mucousy, and it means that their DNA kind of gets trapped in their goo. And we also had a bit of difficulty with red algae because their kind of taxonomic characterizations are very messy, even for visual survey scientists. So at a taxonomic level, it's just a mess. So when we match these sequences to the databases, it's tricky to kind of get direct matches. it's all looking fairly positive. So thank you very much for listening, and if you have any questions, I will take them.
Hey, thank you very much. We'll start here in the row and then over there.
Thank you. That was really interesting. eDNA is fascinating.
It's basically magic, isn't it?
I was just wondering, kind of two-part, how long eDNA stays in the environment, and because obviously yours is all water-based, whether there's likely to be kind of cross-contamination of sites because of the movement of water.
Yeah, there's a lot of work that's coming out that's looking at kind of like those, again, spatial temporal dynamics. A lot of work that looks at the degradation shows that roughly 48 hours is how long marine eDNA hangs about for. That is quite rough, but that is where the kind of ballpoint mark where we're at the moment. And that is because it does degrade quite fast within the marine environment. I have to spend a lot of time and prep making sure that I don't degrade my samples from when I collect them, filter them, and get them back to the lab. So the work that's coming out is showing that the eDNA is quite spatially discrete, even at quite small scales, which is why I kind of hypothesized that my local scale samples are going to be showing some differences. And I still expect that, but I just need to kind of obviously explore the rest of the data set.
I think we have one over on that side. Oh. Right.
Hi, Dina. Great talk. Just following on from that kind of spatial representation issue. With rock pools, obviously, they get replaced with water and tidal cycle. And you've got your distribution sites across the shore, but essentially, aren't they all just samples of the tidal water that have been captured at different time points? So I'm not sure you would actually necessarily expect a difference, because although you've got the rock pool communities contributing, you've obviously got all the species that are in the tidal water, putting DNA into that tidal water, which then fills up the rock pools.
So the whole purpose of setting up this design was that we wanted to know what is the best way to sample a site in terms of maximizing resources and maximizing time. And we did not know whether we could just scoop up some tidal water or whether we could just scoop up some rock pool water and get the same result. Hence why I've decided to do this setup so that I can actually see, obviously the DNA that's going to be in the tidal water is also going to be in the rock pools. But what we want to know is, if we did sample at different points of the shore, will it produce the same results? If it gets implemented in large scale monitoring programs. And if it doesn't, then that means we have to kind of adapt our monitoring strategies to make sure that we're capturing a good representation of the whole site. In an ideal world, we just want to collect 3 samples from the open water because it's so much easier. But that's kind of like what the questions my project is looking to answer.
Can I be really cheeky and answer the question before you go to the number?
Sure, I think we've got time for 2 more, so we'll do you and then someone else.
I just wanted to ask, obviously the kind of water temperatures that you get between kind of South Wales and up in Scotland are going to be very different. Do you think that has an effect on how long DNA is preserved for in the water?
Yeah, so I've collected loads of environmental data as well. I just didn't present it, but yeah, I've collected temperature, pH, rock pool size. I've recorded all of the weather information, and so yeah, exactly. So the environmental variables that influence eDNA are kind of similar to what you would expect for microorganisms to thrive. So if microorganisms are doing well, kind of like in slightly warmer temperatures, it's likely that the DNA is going to degrade faster. And the same for kind of UV as well. So if you've got more UV, again, your DNA is likely to degrade faster. And this work is all still literally coming out over the last 2 years. There's a paper that's kind of looking at those environmental variables. There's only 1 or 2. All of the spatial work is very, very new, and there's not much to go on. So that's why I've tried to collect as much as I could to then kind of figure out what's going on. But, yeah.
Okay, we'll have 1 more question and we'll move on to our last speaker.
This is quite a hypothetical and might not really even be relevant or possible. But I was wondering if there's any way in eDNA to see a difference, I don't know, in like the DNA signal strength or the amount of DNA per species that's there between so that would allow you to differentiate between abundance at all? I didn't know if it was possible.
Yeah, so with metabarcoding, it's really tricky. because the whole lab process, you have to kind of amplify the DNA, and there's multiple different steps where you have to concentrate it to the right amount to get it onto the sequencing machine. So by the time you've done that whole process, the concentration and the read abundance doesn't really mean very much in terms of the true abundance in the environment. You can still look at relative abundance and use it as a proxy, but with a lot of studies have found that there's not much of a relationship, specifically with metabarcoding. But if you're doing single species detection, such as qPCR, you can directly relate the abundance to the single species. So a lot of studies actually do that, but with metabarcoding, it's a whole other gig. I think there's some really, really new work coming out where they spike the DNA at the start with a known amount or concentration and follow that through the pipeline to then kind of mitigate for those different amplification steps, which is really new, and I haven't done that. And it'd be cool to do it, but I think it's pretty nuanced. So, yeah.
Okay, thank you.
Thank you.
A round of applause.